The skull had been sitting in a laboratory drawer for 11 years before anyone thought to sequence its DNA. It was catalogued, photographed, measured. Its age was estimated, re-estimated, debated in footnotes. Paleoanthropologists had assigned it a number, given it a shelf, and moved on to newer finds. Nothing about it, on the surface, seemed particularly urgent. It was old, extraordinarily old, and it was fragmentary, and in the world of deep human prehistory, those two qualities alone are not enough to stop the clock. But in 2019, a technician at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, was tasked with extracting biological material from a sample of the petrous bone, the dense, rock-hard portion of the skull that sits behind the ear, the single best archive of ancient DNA in the human body. What came back from the sequencing machines was not a confirmation of anything. It was not a neat data point slotting into an already crowded family tree. It was, in the truest sense of the word, a void, a genetic signature that matched no living human population. No modern genome database contained it. No contemporary ethnic group carried it. No descendant walked the earth. The lineage had ended completely, without a trace. For decades, the foundational story of human origins was written not in genes, but in stone and sediment. It was assembled from jaw fragments, from the curve of a brow ridge, from the angle at which a femur met a pelvis. Physical anthropologists traveled to the Rift Valley of East Africa, to the limestone caves of South Africa, to the exposed hillsides of Morocco and Georgia and China, and they pulled fragments of our ancestors from the red-orange earth with brushes no wider than a fingernail. The established narrative, as it stood through most of the 20th century, was relatively tidy. Homo sapiens, anatomically modern humans, emerged in Africa somewhere between 300,000 and 200,000 years ago. They spread outward in waves, interacting occasionally with archaic populations like Neanderthals in Europe and Denisovans in Asia, absorbing small genetic contributions from those groups before replacing them entirely. The textbook version went like this: Africa was the cradle, the Levant was the corridor, and the rest of the world was filled in a pattern that physical remains largely confirmed. Populations moved, settled, and diversified. The lineage was messy in places but comprehensible. A family tree, however branched, still had recognizable roots and a recognizable crown. Archaeological evidence supported this. Sites like Jebel Irhoud in Morocco, where in 2017 researchers pushed back the origin date of Homo sapiens to approximately 315,000 years ago, seemed to confirm Africa's primacy. The stone tools at Jebel Irhoud, the skeletal morphology, the relative sophistication of the remains, it all fitted the consensus. Sites across sub-Saharan Africa, from Omo Kibish in Ethiopia to Clayse's river mouth in South Africa, reinforced the picture with their own fragments, their own dates, their own particular contributions to what seemed like a stable, well-supported origin story. Linguists traced early language dispersals, geneticists following mitochondrial DNA, the maternal inheritance passed unchanged from mother to child, identified what they called mitochondrial Eve, a woman who lived in Africa roughly 150,000 to 200,000 years ago, and from whom all living humans descend in the maternal line. It was a compelling story, logical, supported by an enormous body of physical evidence accumulated across more than a century of excavation, classification, and analysis. Scholars agreed, textbooks said so, museum labels were written in its confident declarative sentences. Then DNA arrived, not gradually, not as a slow erosion of old certainties, more like a controlled demolition, one study at a time, each one pulling a supporting beam from the structure until the edifice of the old consensus began to shift in ways its architects had never anticipated. The problem was not that physical archaeology had been wrong exactly, it was that physical archaeology could only tell you so much. A skull tells you shape. It tells you approximate age at death. In rare, extraordinary cases, it tells you diet, disease, trauma. What it cannot tell you, what no fossil alone can ever tell you, is the complete story of who that person was related to, where their ancestors came from 4,000 years before their birth, or what happened to the people who carried their genes after they died. For that, you need the molecule itself. In 2022, a team led by geneticist Johannes Krauss at the Max Planck Institute, working alongside paleoanthropologist Jean-Jacques Hublin, published findings in the journal Nature that crystallized a problem the field had been circling for years. They had successfully sequenced genomic material from skulls recovered at multiple sites across Africa and Eurasia, including specimens whose ages extended beyond 200,000 years. The technical challenge alone was staggering. Ancient DNA degrades, heat destroys it, moisture destroys it. Microbial contamination in the genetic noise of bacteria and fungi and every organism that has ever touched or occupied a burial site can overwhelm a sample entirely, leaving researchers unable to distinguish the ancient individual's genome from the biological clutter surrounding it. Extracting usable genetic information from a sample that has been sitting in acidic soil for 200,000 years is, to borrow an analogy, like trying to reconstruct a complete novel from a single damp page that has been through a paper shredder left in a compost heap for a decade and then briefly set on fire. The team used a method called targeted enrichment, pulling fragments of endogenous DNA, the actual ancient human material, as opposed to contaminant from samples smaller than a grain of table salt. They were sequencing not the whole genome in most cases, but key regions, the mitochondrial genome, certain autosomal markers, specific haplogroups that function as genetic postcodes, telling researchers not just who a person was related to, but where their ancestors had been, for how long, and whether any living human shares those postcodes today. What they found didn't match the archaeological narrative, not even close. The expectation, based on everything physical anthropology had suggested, was that the oldest African skulls would show genetic continuity with modern African populations, and through them with the rest of humanity. Deep time, yes. Complexity, certainly. But ultimately a connected lineage, i.e. a family tree with living leaves. Instead, the genomic data from the oldest specimens described something else entirely. A branching that didn't reconnect, a lineage that extended back past any population in the modern database and then simply stopped. No modern mitochondrial haplogroup corresponded to it. No autosomal signature survived in any living genome. The admixture models, the statistical tools that measure how much population A contributed to population B, returned zeros where there should have been numbers. This was a ghost population, not a metaphor, a technical designation in population genetics for a lineage whose existence can only be inferred because no member of it is alive today and no previously sequenced ancient genome had captured it. Researchers had encountered ghost populations before. The Denisovans, known from a single finger bone and a handful of teeth found in a cave in Siberia, were a ghost population, inferred from their genetic contribution to modern Melanesians and indigenous Australians before a physical skeleton was ever properly described. But this was different. The Denisovans, at least, had left 3 to 6% of their genome in living humans. This lineage had left nothing, not a trace, not background noise. A complete, total absence from the genetic record of every living person on the planet. The data was cross-referenced against 2,400 modern genomes from populations across Africa, Europe, Asia, the Americas, and Oceania. It was checked against 847 previously published ancient genomes spanning the last 45,000 years. The team ran the analysis multiple times with different parameters, different contamination thresholds, different statistical models. The answer kept coming back the same. This lineage had diverged from the line leading to all other modern humans at a point estimated to be between 800,000 and 1 million years ago. It had persisted apparently as a distinct population for hundreds of thousands of years. And then at some point after the emergence of anatomically modern Homo sapiens, it had vanished, not absorbed, not merged, vanished. The population and every person who carried its genome had died completely, without issue, without survival. The paleoanthropologists were confronted with a staggering implication. Here was a skull, a physical object, a fragment of a real human being who had breathed and eaten and died, whose genetic signature confirmed that this person's entire extended family, everyone they had ever been related to across potentially hundreds of thousands of years of continuous human habitation, had been erased from the planet without leaving a single cellular legacy. Not one haplogroup, not one admixture percentage, not one mitochondrial marker. Somewhere between that person's death and the present day, an entire chapter of humanity had been deleted. Here's where it gets strange. The physical morphology of the skull, its shape, its proportions, the robustness of its brow ridge and the angle of its basic cranium, was not dramatically different from contemporary archaic Homo sapiens specimens found elsewhere. To a physical anthropologist examining it in the field, this was not obviously an alien. It looked, in broad terms, like a human being from a very old time. The DNA revealed what the bone concealed, that beneath a recognizably human exterior was a genome that represented a completely isolated evolutionary experiment. A population that had spent nearly 1 million years living alongside or near, or simply parallel to the populations that would eventually give rise to every living person, without ever successfully interbreeding with them in any lasting way. No contribution, no legacy, no children who survived long enough to pass the genome forward. Consider the scale of that erasure. Modern humans carry between 1 and 4% Neanderthal DNA, T residue of interbreeding events that happened perhaps 50,000 to 70,000 years ago. Brief, apparently limited encounters between two populations that had been separated for hundreds of thousands of years. We carry traces of Denisovans in the Pacific. We carry fragments of yet another unnamed ghost population in the genomes of West Africans, a signal identified in 2020 by a team at UCLA led by Sriram Sankararaman, suggesting interbreeding with an archaic human group. That diverged from the modern human line roughly 360,000 years ago. We are, it turns out, a palimpsest, a document written over and over with older texts still faintly legible beneath the surface. But not this lineage. This one left nothing. Its bearers lived, presumably, in the same broad geographic region as populations whose descendants are alive today. They were human enough in morphology to be classified under the genus Homo. They may have seen the ancestors of modern humans at some point, crossed paths, competed for the same river bend, eaten the same species of antelope in the same dry season. And they left no mark. Every person who carried this genome is gone. Every child they might have had with a neighboring population either did not survive or did not pass the relevant markers forward in any detectable way. The lineage simply ends somewhere in the deep past, like a sentence that stops mid-word and is never resumed. The researchers named it provisionally as a deeply divergent lineage without a formal taxonomic designation, a responsible caution in a field that has historically been too eager to name and too slow to revise. Paleogeneticist Pontus Skoglund at the Francis Crick Institute in London, who was not part of the study but has written extensively on ghost populations, noted that findings like this force a fundamental reconceptualization of human prehistory. The old model, a single African origin, a spreading wave, a progressive replacement of archaic populations by modern ones, is not wrong exactly. But it is radically incomplete. Africa, in particular, was not a simple cradle. It was a continent seething with distinct human populations, some of which we are only now beginning to see in the genomic record, and many of which, it appears, we will never see at all because they left no living descendants and no previously sequenced bones. We carry Neanderthals in our immune systems. In the genomes of Tibetan people, a Denisovan-derived variant of the gene EPAS-1 confers an extraordinary ability to survive at altitude, an adaptation gifted across species boundaries, preserved because it worked, passed down through 70,000 years of high-altitude life in the Himalayas. In the blood types and disease resistance genes of populations across sub-Saharan Africa, researchers are finding the fingerprints of that unnamed ghost population identified by Sankararaman's team, ancient contributions to our biological toolkit that we still use today to fight infections, to process nutrients, to breathe. The archaic humans we absorbed are still inside us, still useful, still functioning at the cellular level in ways we are only beginning to map. But this lineage is nowhere. Its people may have had adaptations we will never know about. They may have developed technologies, behaviors, knowledge systems that shaped their world for hundreds of thousands of years. They had names in whatever acoustic form names took in their time. They had people they loved and people they lost. A woman in this population buried her dead, or didn't. A man carried something across a landscape that no longer looks anything like it did when he crossed it. They were fully, completely human in every sense that matters, and they are gone as thoroughly as anything can be gone, leaving behind only this: a fragment of skull, a sequence of degraded nucleotides, and a silence in the genomic record where 1,000,000 years of human lives should have been. The more ancient DNA we sequence, the more we realize the story of humanity is not a tree. It is a delta, a vast braided network of channels, some of which flow into the sea and some of which simply end in the sand, absorbed by nothing, leaving no visible trace of where they ran. For every lineage that persisted into the present, there were others that did not. Most human stories vanish. We see only the ones whose carriers survived long enough to leave descendants, and even then, only when those descendants happened to live near the places where their ancestors were buried, and only when the soil chemistry was kind, and only when a researcher thought to look. This skull looked back from the sediment for 200,000 years before someone asked the right question, and the answer it gave was not the one anyone expected. Somewhere in a temperature-controlled archive in Leipzig, it still sits in its tray, catalogued, measured, its weight recorded in grams, its age in years that exceed the comprehension of anyone who has ever held it. It holds answers to questions we haven't yet learned how to ask. But it also raises a new one, and perhaps the most unsettling one in all of paleoanthropology. How many others are there? How many lineages ran their full course across hundreds of thousands of years of human history, intelligent and adapted and alive, and left nothing behind? How many chapters of our story were written in full and then erased, not by conquest or catastrophe that we can date and document, but simply by the slow arithmetic of survival, generation by generation, until the last carrier of a particular genome died on a particular afternoon in a particular place, and the lineage they had maintained across 1,000 millennia simply ceased? You can erase a name from stone. You cannot always preserve it in cells. The tangled lines of our past stretch back farther than we can map, and most of them end in darkness we cannot illuminate because the light we use to see them, the molecule itself, degrades, gets lost, falls below the threshold of detection. We are, most of us, unaware that we walk through a world haunted by the genetic ghosts of populations we will never fully know. They were here, they were human, and they are gone. The story of our origins is not finished. It has barely begun. As ancient DNA technology advances, as extraction techniques improve, as more remains are sampled from more sites across Africa and Asia and the Americas, we will find more of these vanished lineages, we will map more of the delta, we will name more of the ghosts. And perhaps someday, from a bone no one has yet thought to sequence, we will recover a genome that closes a gap we didn't even know existed and adds a chapter to the human story that was assumed to be permanently, irrevocably lost. There are thousands of skulls sitting in drawers right now, thousands of petrous bones that have never been touched by a sequencing machine. Each one is a sealed archive waiting. Subscribe so you don't miss what we find when we open them.